In this talk, we're going to learn about cytokines. We will learn about how cytokines are produced and the different groups of cytokines as well as chemokines. And finally, we will learn about different functions of cytokines. Now, some proteins and cells that play critical roles in innate immune defenses are always present in the host body. These include proteins like lysozyme, defensins, or complement proteins. Thus, they are constitutively present. We also have cells like monocytes and neutrophils that play crucial roles in innate immune responses, and they are also always present in the host body. However, when there is infection that occurs, then some proteins have to be made in response to that infection. And thus the expression of these proteins have to be induced. Usually these are proteins that are involved in inflammation. And their expression has to be induced because if these proteins are present all the time, or if there is constitutive expression of these proteins, then it is actually detrimental to the host. The host is able to recognize microorganisms that cause infections by recognizing molecular patterns that are present on these pathogens. These patterns are called as pathogen-associated molecular patterns, or PAMPs. The PAMPs are recognized by molecules made by host cells, which are pattern recognition receptors, or PRRs. Now, the pattern recognition receptors are not only able to recognize PAMPs, but they're also able to recognize damage-associated molecular patterns, or DAMPs. And these patterns occur when there is damage to tissues. The pattern recognition receptors can be soluble molecules like ficolin or mannose-binding lectin, but they can also be membrane-bound or intracellular molecules. The pattern recognition receptors that are present in host cells come under different families. Some come under the toll-like receptor or TLR family of proteins, while others come under the NOD-like receptor or NLR family of proteins. We do have pattern recognition receptors that belong to the RIG-1 family of proteins, as well as the STING family of proteins. When pattern recognition receptors that are present in host cells, as shown in the figure, identify PAMPs that are present on pathogens, signaling pathways get activated in the cell. An outcome of the signaling pathway is the activation of a series of proteins, and these proteins, once activated, can activate other proteins. Ultimately, transcription factors get activated, and they can bind to different regulatory sequences on DNA and induce the expression of a variety of genes, like cytokines, chemokines, and other molecules. The cytokines and chemokines that are formed by the cell that is responding to the presence of a PAMP are released in the environment. These cytokines that are released in the environment can mediate their effects on target cells that will have receptors that can sense the presence of the cytokines in the environment. These cytokines and chemokines can mediate physiological processes like inflammation and chemotaxis by binding to their appropriate target cells. Structurally, cytokines are small proteins that are secreted, and they can be produced by a variety of cells. Cytokines bind to specific receptors on cells in order to mediate their effects. And this can be done in an autocrine manner where the cell that produces the cytokine can also express the receptor that binds to the cytokine, allowing the cell to respond to that cytokine. The cytokines can also mediate their effects in a paracrine manner where the cell that makes the cytokine is able to mediate the cytokine effects on nearby cells because the nearby cells now have receptors that can bind to the cytokines. Cytokines can also mediate their effects in an endocrine manner, where the cell that makes the cytokine is quite far away from the cell that actually responds to the cytokine. In such scenarios, the cytokines are carried by circulation to cells that are far away and mediate their effects. There are many groups of cytokines, and some of the main groups include the interleukin-1 family, the hematopoietin superfamily, the interferon family, and the TNF family. Many chemokines are also cytokines, but they mediate chemotaxis, and that is why these type of cytokines are called chemokines. Cytokines mediate many functions and hence can show pleiotropy. In such situations, the same cytokine, like interleukin-6 as shown in the figure, can have different functions in different cells. This pleiotropic effect of cytokines is very important in allowing the host to mount effective immune responses. Cytokines are known to show redundancy, and in this case, different cytokines can mediate the same function. So if we take the example of an activated helper T cell, it can make different types of cytokines like interleukin 2, 4, and 5. And all three cytokines, which are structurally different from one another, can go ahead and act on B lymphocytes and induce the same outcome, which is proliferation. So in this case, all three cytokines mediate the same function and hence are redundant. Cytokines can mediate a wide variety of effects. They can affect the local environment or the site of infection, and they can do many different things like induce inflammation. Pro-inflammatory cytokines are the cytokines that induce inflammation, and they're able to do so by altering the blood vessel properties. This alteration of blood vessel properties results in the promoting of fluid accumulation at the site of infection and also the generation of heat, which are some of the hallmarks of inflammation. While some cytokines are pro-inflammatory in nature, there are other cytokines that are anti-inflammatory in nature, and these cytokines suppress inflammation. There are cytokines that function as chemokines that can promote the migration of cells to the site of infection, and this is done by chemotaxis. There are cytokines that are able to activate immune cells at that site of infection. While there are cytokines that can activate immune cells, we do have cytokines that can inhibit immune cell activity. Thus, cytokines can mediate a wide variety of outcomes. In this figure, we are able to see the effects of 4 cytokines and 1 chemokine. We're able to see local effects that include the localized tissue destruction or increased vascular permeability, as well as chemotaxis of immune cells to the site of infection. All of these are happening in the region where the pathogenic agent is present. Now, these cytokines can also have systemic effects, and systemic effect normally includes the production of fever. Now, fever is an elevated body temperature and plays an important role in helping the host go ahead and destroy pathogens. To look a little bit more into the systemic effects of cytokines, we can observe how some cytokines can influence the liver to start producing more acute phase proteins like the mannose-binding lectins, C-reactive protein, and other proteins that are known to mediate inflammation. The liver is far away from the site of infection, and hence this is an example of a systemic effect of the cytokine. Cytokines can also affect the bone marrow and cause the bone marrow to make more neutrophils and mobilize those neutrophils into the bloodstream. Cytokines can affect the hypothalamus, which maintains the body temperature, and thus it can cause the hypothalamus to increase the body temperature so that we come down with the fever. In order to increase the basal temperature of the body, more energy is required, and thus cytokines can promote the degradation of fat and muscle so that the body temperature is elevated. The fever that is a result of this elevation of body temperature helps in the clearance of pathogens by decreasing viral and bacterial replication, but also increasing some of the host's immune responses. Cytokines can also affect dendritic cells and mediate their migration to lymph nodes, where they can proceed to activate the cells involved in the adaptive immune response. With this, we come to the end of our talk, where we learned about cytokines and different groups of cytokines and chemokines, as well as the function of different cytokines.